Our topic of discussion for the day is protein chemistry. So what is basically protein? Protein is a molecule which is very important for a growing child or for the daily activities to take place. It is one of the most important constituent of our body. It is one of the most important macromolecule which is required by the body. So what is this protein made up of? Protein is made up of a sequence of amino acids. So there we already know that there are basically 20 different types of primary amino acids. Apart from that, there are two more types of amino acids. One is a very newly discovered 21st amino acid, which is selenocysteine, and we have another 22nd amino acid which is also very recently discovered, which is nothing but pyrrolysine. Selenocysteine is very important constituent of glutathione peroxidase or superoxide dismutase, which has got certain properties in the in the ROS or the reactive oxygen species to be scavenged. So now coming to the proteins. So proteins are molecules having a series of amino acid sequence. Based on certain features, proteins can be classified into certain types. We are just basically concentrating on the primary 20 primary universally accepted amino acids. So based on the structures, first of all, protein can be divided into aliphatic, amino acids, aromatic, heterocyclic and derived amino acids. Aliphatic amino acids can be divided into monoamino monocarboxylic acid, which can further be divided into simple amino acids such as glycine, alanine. Glycine is a compound which is an amino acid which has got a chiral carbon, which is has a chiral carbon, whereas the others such as the amide containing amino acid asparagine, glutamine, branch chain amino acids are leucine, valine, isoleucine. The hydroxy containing amino acid are threonine, serine. Sulfur containing are cysteine and methionine. Methionine is an amino acid which do contain a thioether group, and cysteine molecule, when they are conjugated with another cysteine molecule, it forms cysteine. So there is a difference between cysteine and cysteine. Then monoamino dicarboxylic amino acid can be again be classified into two types. One is the glutamic acid and the other is an aspartic acid. Glutamic acid, as we already know, it is one of the most important amino acid which is required in our daily diet for various purposes. So because there are various gamma carboxy glutamated or proteins present in our body. Then coming to the dibasic monocarboxylic acid, which will be lysine and arginine. Lysine has got an epsilon amino group and arginine has got a guanidinium group. These are all the speciality which the proteins have. Amino acid includes proline. Proline is a molecule which has got an amino group have a secondary amine group. Means here in all this we have here we have the amino group attached to the C alpha position, whereas here we have the NH3 molecule which is attached to a secondary carbon, and it is having a cyclic structure type of a structure. Aromatic amino acids is phenylalanine, tyrosine, and to certain extent also we can classify tryptophan as a aromatic amino acid, because these are the amino acids which actually gives the in spectroscopic method when we determine the concentration of protein, these are the aromatic amino acids which shows the quantity or which determines the quantity of protein in the structure. Heterocyclic amino acid will include histidine and tryptophan. Histidine has got a imidazole group, whereas tryptophan has got a indole group. Histidine has got an imidazole group and tryptophan has got an indole group in it. So this is what we have basically. And now coming to the derived amino acid. Derived amino acids are not normally present in the case of the in general in our body structures, but these are present in certain pathways like the citrulline, ornithine are very important, and they are present in the urea cycle, are the major components of the urea cycle. Based on side chain, again amino acids can be classified into non-polar, charged and uncharged. Non-polar side chain includes leucine, isoleucine, valine, alanine, methionine, proline, etc. Whereas the charged can be again divided into basic and acidic. Basic includes the lysine, arginine, histidine, whereas the acidic includes the aspartic acid, glutamic acid. The uncharged or the non-ionic includes the glycine, serine, cysteine, threonine, tyrosine, glutamic acid, and also glutamine and also asparagine. Based on the metabolism, it can be classified into ketogenic, which includes leucine, ketogenic and glucogenic, includes lysine, isoleucine, phenylalanine, tyrosine, tryptophan, and all the other 14 amino acids are glucogenic in nature. So what is basically a ketogenic amino acid? Amino acid which can be converted to acetyl CoA and can enter the ketogenic pathway is known as a ketogenic amino acid. Whereas a glucogenic amino acid is a protein molecule, or it's an amino acid molecule which can be converted into a substrate which can act as a glycolytic intermediate or can enter the glycolysis pathway. So this is the based on the metabolism. Now coming to based on the nutritional requirement, it can be classified into essential, non-essential and semi-essential. Essential amino acids are the amino acids which are not at all present and which are very much required in the diet, and so we need supplementation for these essential amino acids. Non-essential amino acids are the amino acids which are not at all required because they are constantly being synthesized inside the liver, so we don't require this amino acid at all in the diet. And we have a semi-essential amino acid which is required in small amount in children, and in adult as the life progresses we do not require it anymore. So the semi-essential amino acid will include mostly the arginine and histidine. So this is more about the classification of proteins. Now let's come to the properties of proteins. Proteins can be classified as amphoteric because it has both acidic and a basic component. The amino acids basically is something like so here basically we can say, so here we are having a cationic part and an anionic part. So it's actually in the neutral pH or in a zwitterionic state, it is neutral. Whereas in the case when it is moved towards a pH which is lower pH, so in low pH it is basically cationic, meaning it is present as COOH, CH, NH3 plus and R. In the zwitterionic state, it is present, it is having both, and so it's called the amphoteric in nature. Whereas at higher pH or at an alkaline pH, we see that it has got, it is mostly in its anionic state, and so it is having COO minus, CNH2HR. So this is the anionic state at a high pH. So this is the one of the most important property of proteins, which is a zwitterionic state. Now the proteins usually are present in the L form, unlike the carbohydrate molecules which are present in its D form. So first property is zwitterionic state. Number 2 property is is optical activity. Here we will see that all the amino acids are basically seen in its L form. So all the amino acids mostly are seen to be present in its L form, unlike the carbohydrates which are present more prevalently in its T form. Now the next property will be the peptide bond. What is a peptide bond? Peptide bond is basically a linkage, or it's a chemical bond which is formed between the COOH group of one amino acid and the NH2 group of the other amino acid, or the second amino acid. So it is basically so it is somewhat like this, and this is the structure of a, this is the peptide bond. This is a peptide bond. It is a nothing but a CONH bond, where the subsequent, this is a peptide bond. This is also called the amide bond. It is a partial double bond because it is having a bond length which is intermediate between the primary and the secondary, or rather the single bond and double bond. So it is having a bond length of about 1.32 angstrom. So it is having a bond length which is intermediate between the single bond and double bond. So it is having a partial double bond character. All the 6 molecules are present in the same plane. So it is a partial double bond. All the molecules are in the same plane. We have already discussed how it is formed. Secondly, it can be that this peptide bond which is present between the 2 molecules can therefore be this peptide bond is a very important structure which actually gets denatured in the case of denaturation of protein. Moreover, this CONH bond, we will see that this R group in the subsequent amino acids, when they are joined, they will be in the trans form. So the amino acids are basically produced, or they are attached, having their R group in subsequent or adjacent amino acids present on 2 different sides. This is so they are present in the trans form, so that there is decreased amount of steric hindrance. Now coming to the next part, which is the peptide bond, we have completed. Now the next part which can be discussed is stability. That is, the protein molecules such as alanine, valine, isoleucine, these structures have a capability to basically stabilize a protein structure. Molecules like valine, leucine, isoleucine stabilizes the protein structures, whereas molecules like proline will disrupt the structures. So if proline is present, it is going to disrupt the structure. So the properties of proteins will include the zwitterionic state, or amphoteric or amphibolic in nature. It is going to have an optical activity, peptide bond formation, which is basically is present in the trans form, and we also will be discussing about something called the Ramachandran plot. Ramachandran plot is basically a representation, it's rather a graph In this Ramachandran plot, we are going to discuss, the Ramachandran plot basically describes the structures which are rather stable structures of the protein, which are more favorable structures. This is basically, we don't need it in too much details, so we are just going to do this briefing that there is something called the torsional angle. So based on the torsional angle, the protein molecules are depicted in a graph, so that we can, so that the various classifications, or by seeing a graph we can depict whether some protein is stable or not, or whether it's present in a stable form or not. So this torsional angle is basically dependent on two types of angle. One is the phi angle and one is the psi angle. So there are two angles, the phi and psi, which are known as the torsional angles, which will basically depict the Ramachandran plot, which is a structure, it's a graph which represents the protein molecules which is stable. Now we have done with the properties of the protein molecules. Now we will be going towards the reactions of proteins. Reactions of proteins. First are the reactions which are concerned with the amino groups. The first one which we will be discussing about is nothing but transamination reaction. Transamination reaction is nothing but the transfer of an amino group from a keto acid to a from an amino acid to a keto acid, thus leading to the formation of a new amino acid. Suppose we will be discussing about two different reactions. One is alanine plus alpha ketoglutarate. It will give pyruvate plus glutamate. So this is a very important reactions which we have, which is alanine plus alpha ketoglutarate gives pyruvate plus glutamate. Here we use ALT, or alanine aminotransferase. We use alanine aminotransferase. Another very important reaction is that where we use oxaloacetate plus glutamate gives alpha ketoglutarate plus aspartic acid. It is the same reaction. Here we use another reaction where we use another enzyme. This is also a transaminase. So this reaction, basically the transamination reaction, is a reaction which requires PLP. PLP, which is pyridoxal 5' phosphate. Pyridoxal 5' phosphate is a derivative which is derived from pyridoxine. So PLP is actually present. This PLP is attached to the enzyme molecule by a Schiff's base imine linkage. So it is attached to this enzyme molecule by a Schiff's base imine linkage. These two reactions which we have already discussed in the transamination reaction are very important because they are the very primary reactions which takes place inside the liver and which decides whether your liver is working properly. These reactions, which we have already discussed, ALT and GPT, the two enzymes inside the blood are classified as the serum SGOT and serum SGPT, which is serum glutamate oxaloacetate transferase and serum glutamate pyruvate transferase or transaminase, whatever it may be. These two enzymes, if they are elevated in the blood, determines liver failure. Therefore, these two enzymes, the transamination reaction is so very important in our body because it is a biomarker for liver functioning. So this is what we have for transamination reaction. Now moving to the next part, that is the oxidative deamination. Oxidative deamination takes place by the help of enzyme glutamate dehydrogenase. It is basically a mitochondrial enzyme present, and it is only being the reaction only occurs in an anaerobic environment. So this oxidative deamination can be classified as a glutamate dehydrogenase dependent, and it is also taking place in an anaerobic environment. It is an enzyme which is present in the mitochondria, and also that it requires NADP. So this is a very important nicotinamide adenine dinucleotide phosphate is an important requirement for this reaction. Here we will see that glutamate dehydrogenase will lead to the formation of a product initially this is alpha aminoglutarate, and finally this will give rise to nothing but alpha ketoglutarate. So first we see glutamate reacting upon with NADP. Here we will see a water molecule is entering, and it will give rise to finally NADPH plus H plus. So COO, and so glutamate molecule reacts with NADP, glutamate dehydrogenase, forms alpha aminoglutarate. This is a transient form, or it's an intermediate, which finally leads to the formation of alpha ketoglutarate, and finally giving rise to NADPH and H plus molecule. So this is what is the very important step of oxidative deamination. So here also we are basically also getting one molecule of NH4, which is being released. This NH4 will then enter the urea cycle. So this is the basic structure of the oxidative deamination. We also have another type of oxidative deamination, which is by the reactions by oxidase, where we will be seeing we also have a second type of with deamination reaction, oxidative deamination reaction. Oxidative deamination with oxidase. Oxidase is an enzyme which can be D amino acid oxidase or L amino acid oxidase. The D amino acid oxidase requires FAD as a coenzyme. D amino acid oxidase requires FAD as a coenzyme, whereas the L amino acid oxidase requires FMN or flavine mononucleotide, and D amino acid requires flavine adenodinucleotide as a cofactor or coenzyme for the reaction to take place. Suppose, for example, serine molecule is there, where it's acted upon by D-amino acid oxidase. We get pyruvate. Here we will see that it is getting FAD is converted to FADH2, and then subsequently by the enzyme H2O2 plus O2 by the enzyme catalase. So there is a role of, so this is basically the reaction which is taking place, serine to pyruvate which is taking place through an intermediate step, which is again an alpha amino group. So it's again an intermediate step, and then finally we get pyruvate, where FAD and FADH2 is getting converted, and we are getting here also we are getting releasing one molecule of NH4. So this is the reactions of oxidative deamination, which is taking place with the help of flavin dinucleotide and flavin mononucleotides, which are basically the D and L amino oxidases, which is very important. Now coming to the next part, which is the reactions which do not require, or the reactions which do not require which are also reactions of the amino group, but they do not so they are basically the dehydratases and desulfidrases. So reactions by dehydratase and desulfidrase, here we are not releasing molecules as H+. Rather, we eliminate the eliminating compound which gets eliminated is in the form of either H2S or in the form of water. So it is not directly H+. So here these are the reactions which are basically performed by the dehydratases and the desulfidrases. So the reactions are almost the same thing, where threonine is also reduced to the corresponding keto acids, and also serine is also serine with the help of serine dehydratase will lead to the form alpha amino acrylate, and then this will finally lead to the formation of pyruvate. So this is the reaction which we follow for the normal desulfidrases, and in this step we finally release one molecule of H2S. So a molecule of H2S, sorry, not H2S, a molecule of H2O is released. So this is what we have for the dehydratases, desulfirases. Here lightly we can have serine, we can the compound which can undergo this reaction is serine, cysteine, and also threonine can undergo this kind of a reaction. All amino acids cannot undergo this dehydratase, desulfirase reactions. Now coming to the next part, that is the reactions of the carboxyl group. Carboxyl group. Before coming to that, let's discuss something about the carbamino compounds. That is, the NH2 group of the amino acid can also react with a COOH group to form a carbamino acids, like in the case of carbamino hemoglobin. So the carbamino hemoglobin is basically formed by the reaction of CO2 with the NH2 group or the amino group of a polypeptide chain. So this is what we have for the reactions of proteins. So we have discussed in amino acids the oxidative deamination, transamination, and also we have discussed this dehydratase and desulfidrase reactions. Now coming to the next part, which is the reactions of the carboxyl group. The carboxyl group in other place can undergo the reactions such as the esterification, it can undergo the esterification reaction, where the serine and the threonine group, the COH group in serine molecules, can undergo an esterification reactions. So that is one of the most important reactions which is taking place. Beyond that, there is also certain other such as there is the glycosylation reactions which takes place to form glycoproteins. These are also the various reactions which is where we can form the glycoproteins can be formed. So these are the reactions where COOH group is involved. Another reaction of amino acids which we have forgotten to discuss is the decarboxylase reaction. Decarboxylase reaction, where basically amino acids can be transformed into the corresponding, such as tyrosine gives rise to tyrosine is giving rise to tryptamine, histidine giving rise to histamine, subsequently like this. So carboxylase is an enzyme which is taking place, and so here we will see tyrosine is when acted upon by decarboxylase, it is giving rise to tryptamine, rather tyramine. Tryptophan, when acted upon by the decarboxylase, gives rise to tryptamine. Histidine, when acted by decarboxylase, gives histamine, and subsequently. So this is what we have for the decarboxylase enzymes. So this is what we have now, and so this is all for the what we have for the reactions of proteins. Now we have another reactions of the reactive group, or the R group. Reactions of the R group, which is also quite important. And why on the R group? Rather, we can classify it as the transmethylation reactions. Better to classify it as a transmethylation reaction. Transmethylation reaction is a reaction where we will see that there are several molecules like biotin, S-adenosyl methionine. There are various compounds which acts as a methyl group donor. So these molecules basically will donate the methyl group and itself get reduced. So this S-adenosyl methionine is the most commonly used methyl group donor, which is used in all the biological reactions. And so this methyl group donor, S-adenosyl methionine, after transporting or after giving off its methyl group, will lead to the formation of S-adenosyl homocysteine. So this is what we have. Transmethylation reaction is one of the most important reactions in our body, because the reaction starts with folate. Folate acted upon by tetrahydrofolate gives rise to tetrahydrofolate reductase, gives rise to tetrahydrofolate, which on here we give serine hydroxymethyl transferase, an enzyme where serine molecule is getting converted to glycine, and here we will be getting N5, N10 methylene tetrahydrofolate. So folate is getting reduced by tetrahydrofolate reductase to form tetrahydrofolate. Tetrahydrofolate acted upon by serine hydroxymethyl transferase, serine one molecule of serine getting transformed into glycine, and we finally get N5N10 methylene tetrahydrofolase. This N5N10 methylene tetrahydrofolate is important because this will enter the nucleotide biosynthesis, a very important precursor for nucleotide biosynthesis. And this one thing more to be remembered, that the drugs which we use for cancer, the most cancer drugs basically target the enzyme, the tetrahydrofolate reductase. Drugs such as methotrexate, which are very commonly used for anti-cancer therapy, is one which will inhibit the tetrahydrofolate reductase, and therefore it is going to inhibit the pathway. So we must remember that the methylation reactions are very, very important, because this is one of the examples where methotrexate is being used to inhibit this pathway, because it is an antiviral, anti-cancer drug which is most commonly used. Now, one more thing you have to keep in mind, there are various products like the creatinine molecule, which is formed from guanidinoacetate. It is formed from guanidinoacetate by the transmethylation reaction. So these are the very important compounds which are very much required in our body, which are formed by the transmethylation reaction. Moreover, there are certain molecules which get activated after transmethylation. Even there are certain transmethylation reactions where the steroid hormones, the epinephrine, norepinephrine are being synthesized, where we undergo transmethylation reactions. So transmethylation reaction overall is a very, very important process. So we mostly discuss with respect to SAM, which is nothing but S-adenosine which gets finally reduced to form S-adenosine homocysteine after giving off its methyl group. So this is what we have moreover for the reactions of proteins. So this is what we have for the reactions of protein. Now one more thing which is important is the tests of protein. Here we are just going to discuss about one test of protein, which is the in general test for detections of protein. Test for protein. Boiling can be a very good test for protein, where we are checking for the coagulation. We are checking for coagulation. So boiling is a very good test for protein, because generally at a temperature of about 110 or 120, proteins tends to show to coagulate, property of coagulation. So coagulation can be by just giving heat to a protein molecule. Subsequently, we can see that we have got a Ninhydrin test. Ninhydrin test is very important. This is the one of the most important and the mostly used tests, where we are able to detect the alpha amino acids. So this Ninhydrin test is one of the very, very important things which is present in the case of which is almost shows positive test for all the protein molecules. And lastly, we can also detect the amino acid molecules with the help of another called the spectroscopy, where we use spectroscopic technique. Basically they will we use is the spectroscopic determination of protein molecules basically occurs due to the presence of aromatic amino acids like tryptophan, tyrosine and phenylalanine. So they show a spectroscopic result because they absorb the UV light at a nanometer of about 280 nanometers. So protein shows UV absorption spectroscopy at about 280 nanometers. So how to differentiate between a protein and a DNA sample? DNA in general has the tendency to show absorbance at about 260 nanometers. So there is a slight difference between protein and amino acid proteins and DNA molecules, which helps to separate proteins from a DNA sample. So this is how we can separate the protein and the DNA sample. Protein molecules shows absorbance at about 280 nanometers due to the presence of the various amino acids like the tyrosine, phenylalanine and tryptophan. One more thing I can tell, I forgot to mention previously, that is that the protein molecules basically has two ends. One is a carboxyl end, other is the amino end or the nitrogen containing end. So that ends are also very much important for the various numberings of the amino acids. So this is what we have for the tests of protein. Now coming to the structures of the protein. Protein structure is very important because we know that the protein can exist in several structures. First, let's come to the primary structures of protein. Primary structures of protein is basically nothing but the chain of amino acid. It is a normal chain of amino acids where we have some amino acids present such as lysine, serine, etc. Whereas in the secondary amino acids we have some hydrogen bond present between the primary structures. We have secondary structure will have hydrogen bond present between them. So it is having some hydrogen bond present, and therefore these structures having various shapes, such as it can give rise to an alpha helix or a beta pleated sheet. So these are the various structures which can be present, an alpha helix structure or a beta pleated sheet. Alpha helix structure is a structure which is mostly present in the case of proteins. So the right-handed helical structure having 3.6 amino acids per turn. So similarly we have another structure called beta pleated sheet. Now coming to the tertiary structure. Here the structure is further stabilized by some other bonds, such as the van der Waals forces, such as the hydrophobic reaction, hydrophobic bonds, electrostatic attractions, and several other bonds. So this tertiary structure of protein comes into play like that. And the finally which we have is the quaternary structure, where we have several chains of polypeptides which are attached to one another, giving rise to various subunits of a protein, such as the hemoglobin molecule, which is having 4 subunits, is basically a quaternary protein. So we are having quaternary structure for a protein. So this quaternary protein, basically hemoglobin molecule has got 2 alpha and 2 beta chains. If there is even a slight mutation in any of the genes, it can lead to some frame shift mutation, where it will be giving rise to some hereditary genetic diseases like the thalassemia, and also may lead to even a mutation where there is a suppose in the 6th position, if glutamate is replaced by valine is replaced by glutamate, we see that it's giving rise to sickle cell anemia. So these are the various structures which are various reactions which basically takes place in the hemoglobin molecules. So this is what we have for the protein structures. One more thing to remember is that during denaturation of reactions, denaturation of proteins which we do, which is a very important property of protein molecules, the primary structure remains unaffected. It is only the secondary, tertiary structures which gets damaged. Primary structure of the proteins remains unaffected, or they remain undamaged. So proteins, basically denaturation of proteins is partially helpful, because suppose the proteins which we eat, basically when we are cooking it, actually the proteins gets denatured. This denaturation actually makes the protein more susceptible to enzyme actions which are present in our gut and in the stomach and various other intestinal organs. Therefore, it will lead to the easier digestion of the protein molecules. So for protein digestion, denaturation is quite helpful. So there is a positive aspect of denaturation. So there are various processes by which we can denature a protein. So the denaturation of protein can denaturation of proteins which involves only the secondary and tertiary structure. The primary structures are not at all involved. In the denaturation of proteins, we will see that it can be done simply by boiling. It can be done by some heavy metals like lead, mercury, can be used. Also some salts, which is called the salting out, where we use basically 22% Na2SO4 or NH4 to ammonium sulfate is used. So these are the various salting out, heavy metals, boiling. We can use acids, organic solvents, etc. For acids such as hydrochloric acid is one of the very important acids which is being used. Even hydrochloric acid is also present in the stomach, which is also having some proteolytic activity, as we have already known. And so these are the various structures various compounds which are most commonly used for the denaturation of protein, keeping the primary structures unaffected. In certain cases, we have seen that we can also regenerate the protein back after the removal of the agent which is actually causing the depolarization. And this boiling, which is basically, the boiling is one of the best examples which we see most commonly in our daily life or in everyday life. That is the boiling denaturation, which basically takes place when we boil an egg. An egg being boiled, basically the albumin protein which is present in the albumin protein is getting coagulated. Whatever meat we make, maybe it be the omelette or a fried egg, whatever, it's basically nothing but the coagulation of the protein molecules which is taking place by the process of boiling. So this is what we have for the denaturation of proteins. And now the last thing which we have after the denaturation is some additional facts about the protein molecules. Protein molecules basically are there in every aspect. It's a molecule which is required by almost everything. All the enzymes which are present inside our body are basically protein. Protein is a very important structural component. Structural component like it is a very important component of collagen. Collagen is a protein which is basically a triple helical structure where we have got glycine present in every third amino acid, and the predominant amino acid is a hydroxylysine and hydroxylysine and hydroxyproline. These two are very important amino acids which are present, and this hydroxylysine and hydroxyproline are formed due to the presence of vitamin C. So vitamin C also plays a very important role in the formation of the collagen molecule. Then it is also present in various enzymes. Whatever enzymes we talk about is basically protein, maybe carbonic anhydrase or pepsin, all are protein molecules. Then it is also used in various cell signaling pathways. It is also very important. Means why, what not? Because it is the very various proteins. It helps to maintain the osmolarity, such as which are the plasma proteins. It is also helping in blood clotting. The clotting factors, they are also protein. The antibodies, the immunoglobulins, they are also proteins. So antibodies, IgG, IgM, plasma proteins, maintains osmolarity, and then we have clotting factors. So these are all the various things. Even we have proteins required for cell signaling pathway, cell signaling cascade, and so on. There are so many functions of the protein molecules. It is also, so protein has got no end. So these are the basic things these are some of the very important aspects of various uses of protein molecules. And last thing is that the one of the most important process to detect the protein is by the turbidometry method, which we have already discussed, that it is the method by which actually we can boil the protein and see how much it is getting turbid or how much there is coagulation. Similarly, some deficiency disease caused by protein malnutrition can be kwashiorkor and marasmus, which occurs basically in children. Protein deficiency usually is rare in the case of adults. So this is what we have moreover for protein chemistry. So I would like to thank, and sorry for the disturbances in the background, and hope you liked it, and please do subscribe the channel. Thank you.